Okay, good morning, everybody. Um, so I think we talked about the fatty acids oxidation, right? And then ketogenesis last class. And just wanted to briefly, go over it again. So um, under the fasting conditions, because we don't have enough energy substrate, right, particularly glucose from the diet. So our body started to mobilize stored energy. Uh, Glycogen, of course, it's break down, right, to release into the circulation to raise the blood glucose level. And also our body is able to make glucose from non-sugar precursors, right? We have a list of precursors for gluconeogenesis. And in the meantime, and actually most importantly, the fat tissue, the triglyceride, the stored energy, right, also need to be mobilized or break down to the fatty acids. And fatty acids released into circulation from the fat tissue and picked up by other tissues, particularly muscle. If you still remember, about 70%, right, 2/3 of the fatty acids are released from fat tissue picked up by muscle and utilized as energy during the fasting condition. And 1/3 of the fatty acids that are released from fat tissue actually goes to the liver for fatty acid oxidation and gluconeogenesis. And I wanted to mention you again, in the skeletal muscle during the fasting condition, the skeletal muscle be able to completely oxidize fatty acids, like break down fatty acids to acetyl-CoA, and then acetyl-CoA enter the TC cycle for complete oxidation, and to make ATPs in the muscle. But in the liver break down the fatty acids to acetyl-CoA, and majority of acetyl-CoA will not enter the TC cycle for complete oxidation, rather actually for the ketogenesis, right? Remember this. So, and we also talked about the ketone bodies and be synthesized from acetyl-CoA in the liver. So liver is a major site for ketogenesis. And liver then released the ketone bodies once it's made into the circulation, and then the other tissues, we pick up for utilization as an energy. And I also mentioned that the reason why our body need to make ketone bodies instead of just directly utilize fatty acids, right? Because of the brain, fatty acids molecules too larger, so brain cannot uptake the fatty acids. So fatty acid has to be break down and make a relatively small ketone bodies, and ketone body be able to pass through the blood brain barrier, right, and then be able to enter the brain and utilize for energy. And we also talk about the liver. Also, it makes ketone bodies, but liver cannot utilize ketone bodies, right? So the next slide, I'm going to talk about the utilization of ketone bodies. Again, it's made like acetoacetate and hydroxybutyrate. These are the two common ketone bodies because they are present at relatively high levels in the circulation. So then tissues, particularly muscle, right? Here just as muscle as an example. So muscle picks up ketone bodies and then oxidize, right? So this sort of the pathways how muscles utilize ketone bodies as energy for energy. So hydroxybutyrate can be converted back to acetoacetate, and acetoacetate can be can be converted to acetyl-CoA, right? And this is enzyme called the succinyl-CoA transferase, right? So succinyl-CoA transferase is the key enzyme for the utilization of ketone bodies. So ketone body can be converted to acetyl-CoA, and silase is the enzyme that breaks down acetyl-CoA back to acetyl-CoA, right? So acetyl-CoA eventually enters the TC cycle for ATP production and for complete oxidation. So this is a process for ketone body utilization. So again, the succinyl-CoA transferase is the key enzyme for utilization of ketone bodies. So now the reason why liver cannot utilize ketone bodies because liver actually lacks this key enzyme for ketone body utilization, right? So liver lacks a succinyl-CoA transferase, so liver is not able to break down the ketone body and utilize ketone body, right? Okay, so just the kind of energy yield, I mean ATP production from palmitate. If we make a comparison between the complete oxidation, fatty acids versus and the beta oxidation and ketogenesis, and then oxidation. So see how many ATPs made in these two different paths, the pathway of the fatty acid utilization. So we already calculated before, right? If we let's say in the muscle, if we completely oxidize palmitate to acetyl-CoA and 8 molecules of acetyl-CoA enter TC cycle, and for complete oxidation and generating 129 ATPs, right? And if palmitate is not completely oxidized, first of all, palmitate will go through the beta oxidation, right, and to break down to acetyl-CoA molecules. So if this is 8 acetyl-CoA molecules are not completely oxidized, through the TC cycles, rather they go to the ketogenic pathway, for example, in the liver, right? So 8 acetyl-CoA molecules are used to make ketone bodies, 4 ketone bodies, right? And ketone bodies eventually can enter the TC cycle, right, and break down back to the 8 acetyl-CoA molecules. But because of the, remember that key reactions in the utilization by succinyl-CoA transferase, this reaction utilizes 2 ATPs, right? Sorry, 1 ATPs. So, and for ketone bodies and utilized in the tissues that actually also utilize 4 ATPs. So you, for complete oxidation, you generate 129 ATPs, but because of you use 4 ATPs, so you have a less, ATP being generated. If fatty acids go beta oxidation, ketogenesis, and then ketone body oxidation pathway, right? So less ATP made. But just because of the reason, again, brain cannot utilize fatty acid directly, so we have to, our body has to go through this fatty acid oxidation, ketogenesis, and acetyl-CoA oxidation pathway for making ATPs. Okay, now let's talk about the regulation of ketogenesis. So remember, the ketogenesis occurs during the fasting, right? When we have a lot of fatty acids being released from fat tissue, from triglyceride breakdown. So the circulating fatty acids level is increased, right, during the fasting, particularly, long, prolonged fasting. So fatty acids level increase is very important. That can promote ketogenesis. So one regulatory point is the circulating fatty acids level. So increased fatty acids level, increased lipolysis and fatty acids level is one important regulatory factor. And the second, fatty acids, we know that if it can be oxidized in the mitochondria, it has to be transported into the mitochondria, right? Remember this regulatory point is that the CPT1, right, is a key enzyme that control fatty acid transport from cytosol into the mitochondria. So CPT activity, this enzyme activity is also very critical. If the enzyme activity is increased during the fasting, this enzyme actually can be activated, can be increased during the fasting, so then can promote more fatty acids enter the mitochondria for beta oxidation. And lastly, the acetyl-CoA levels, right, is also very important. This is a substrate for ketogenesis. So acetyl-CoA levels also regulates the ketogenesis, right? So try to remember three important factors that can control or regulate the ketogenesis, the rate of ketogenesis. First is the circulating fatty acids level. And second, the transporter, the regulation, the CPT1, the key enzyme that control the fatty acid transport, right, from cytosol into the mitochondria. And lastly, the acetyl-CoA levels inside of mitochondria is also the key regulatory point. Okay, so ketoacidosis. We know that the ketogenesis, we just talked about it, is considered like under the normal physiological condition, right? Like under fasting condition. So this type of ketogenesis, we also call a dietary ketogenesis, which occurred under the normal physiological condition, fasting condition. But ketogenesis can also happen during the pathological conditions. So the ketoacidosis, that means we have extreme or uncontrolled ketogenesis. The ketogenesis is much higher, a rate much higher than the dietary ketogenesis, right? And so body generate the very high concentration ketone bodies. So and this ketoacidosis most common in uncontrolled diabetes or prolonged alcoholism, right? So diabetic ketoacidosis, like diabetic patient, because of the lipolysis somehow is uncontrolled. So a diabetic patient usually have a higher lipolytic activity. So lipolysis actually increase, so release a lot of fatty acids into the circulation. So again, the diabetic patient, type 2 diabetes, right? Diabetic patient usually have high levels of free fatty acids. So this again we know is one of the important regulatory factor, right? So have a high fatty acids that can be picked up by the liver for making ketone bodies. And alcoholic ketoacidosis, because of alcohol consumption, sometimes can inhibit the gluconeogenesis. So you have less glucose, made in the body, so that will promote fatty acid utilization. So again, also causing the fat mobilization and increase fatty acid level in the circulation. So both sort of pathologic conditions can also cause the extreme or uncontrolled ketogenesis, right? And raise high levels, raise the ketone body levels in the circulation. So here's sort of the general mechanism or pictures of ketoacidosis. So I just mentioned the diabetic conditions or alcoholism. Both conditions, you have a problem with glucose utilization. Alcoholism I already mentioned, right? Because alcohol metabolism can inhibit gluconeogenesis, so you have less glucose available for body use, particularly under the fasting condition, right? And diabetic patients had increased lipolysis and also have a problem with the glucose uptake, right? So you have not much glucose being uptake into the cell, so cell inside of cell actually glucose levels are low. Although you have a high glucose level in the circulation, but inside of cell glucose level is low because of uptake is decreasing. That's problems, right? So in general, under these two pathological conditions, glucose utilization inside of cells actually decrease or lower. So this will again promote the fatty acids oxidation, promote the triglyceride mobilization and fatty acids oxidation. So that will generate a lot of acetyl-CoA and produces again the lots of ketone bodies. Any question about ketoacidosis? Okay. It's just a kind of under the alcoholism condition that the alcohol can block the gluconeogenesis. The mechanism is alcohol can be metabolized to acetyl-CoA, right? Itself can increase acetyl-CoA concentration, but meantime, because there's two reactions, requires the NAD. So NAD, so alcohol metabolism needs more NAD. So NAD is produced from these two reactions. So they utilize a lot of oxaloacetate, right? So oxaloacetate, is part of gluconeogenesis. It's an intermediate of gluconeogenesis, right? So if alcohol, oxaloacetate is depleted by alcohol metabolism, then you have less glucose being made, right? So this is sort of the pathway or mechanism how alcohol metabolism can deplete the oxaloacetate and inhibits gluconeogenesis. Okay, so and I guess this is probably the last two slide, right, for this section. So we're just going to talk about the regulation of a long-chain fatty acid oxidation. So how fatty acids oxidation process is regulated during the fat and fasting condition. So I think this slide is very good, summarize all this regulatory process. So try to remember this, two slides, right? So I highlight the steps with the blue represent the under fasting condition, and red mean that in the fat condition, right? So let's look at in the fat condition first. Yes?
I just have something to say. None of us had the last slide about alcohol, so if you wouldn't mind posting that. The 39 slide. We have this like 2 times.
Do you have this? 2 times? Yeah, the same actually. I'm sorry, actually the same slide, but just I guess, let me see. Yeah,
Oh, like go back one.
to go back one.
Yeah.
Oh, you mean the this one? Oh, okay, yeah, sure, I can.
Okay, thank you.
Yeah. I just made it, 10 minutes ago. um, so this under the fat condition, right? So when we have a high blood glucose level, right, after meal, and this will glucose will be oxidized through glycolysis and to the pyruvate if it's under the fat condition, right? So it should be further oxidized. So then pyruvate enter the mitochondria and converted to acetyl-CoA, right? Remember this step? And by called the enzyme. Still remember the enzyme that converts? Sorry? Yeah, pyruvate dehydrogenase, right? Complex. So, and then generating acetyl-CoA, right, from the glucose under the fat condition. And if you still remember that under fat condition, glucose can be converted to fatty acids, right? This process called lipogenesis. So acetyl-CoA being, and remember this is the first key reaction, right? The lipogenesis. So acetyl-CoA is converted to malonyl-CoA, and by the key enzyme complex, remember, ACC, right? Acetyl-CoA carboxylase complex. And remember, try to remember, malonyl-CoA is also the key molecule. It's a key substrate. Malonyl-CoA can inhibit CPT1 enzyme activity. CPT1 is on the mitochondrial membrane, right? This is a key, again, enzyme that control fatty acid transport from cytosol into the mitochondria. So, so under the fat conditions, because we have lots of glucose, yeah.
How can it inhibit the CPT1? Just by not being there, it's inhibited, or what are the methods that malonyl-CoA uses to inhibit CPT1?
Right, inhibits CPT1.
By not being there, it's inhibited the process, or?
Oh, it's that enzyme is always there, but the activities is being inhibited under the fat condition when we have a lot of malonyl-CoA generated from acetyl-CoA, right? So and we also know that under fat condition, insulin level is high, right? So insulin is, if you still remember, is the important regulators of ACC enzyme. So insulin basically stimulates ACC activity, right? So once this is, ACC activities increase, that will lead to increase the formation of malonyl-CoA, right? Does that make sense? So under the fat condition, when we have high glucose, high insulin, and high ACC activities, the malonyl-CoA formation is increased. So malonyl-CoA level should be high, right, under the fat condition, and this will inhibit the CPT1 activities. So CPT1 we know is a key enzyme that helps fatty acids, right, transport into the mitochondria. So if this enzyme activity is low, then fatty acids transport will be decreased. So you have less fatty acids transport into the into mitochondria. So fatty acid oxidation is decreased under the fat condition, which make sense, right? If we're thinking about comprehensively and big picture. So under the fat condition, we utilize glucose because glucose are available from the diet, so we don't need to break down fat triglyceride, don't need to utilize fatty acids. So fatty acid utilization need to be decreased. So this under the fat condition, so you see all the process and how the key regulate point, right? So try again, remember the marino-CoA, is a key substrate that can control fatty acid movement and oxidation, right? So you have again, have a lower CPT1 activity and lower beta oxidation and lower ketone body synthesis, right? Right? Any questions? Right? Okay, so now let's look at the fasting condition. So fasting condition, we have a decreased glucose, right? So we need to mobilize fatty acids from triglycerides through the lipolysis process. So fatty acids level in the circulation should be high, right? So also thinking about, some substrate or nutrient levels in the circulation in the fasting condition. So let me just ask you, under the fat condition, what about the glucose levels in the circulation? It should be high, right? And what about fatty acids and ketone bodies levels? Should be low, yes. And under fasting condition, glucose levels low and fatty acid ketone bodies level high, right? Okay, that makes sense. So, and fasting conditions, fatty acids levels increase, so acyl-CoA levels are high. And also because during the fasting, the hormone glucagon level is high, right? So, and if you still remember when we talk about lipogenesis, why high-fat diet inhibits the lipogenesis, and we mentioned that long-chain fatty acyl-CoA levels, right? So it's very important when we have a high acyl-CoA. It's the same thing, right? Acyl-CoA or fatty acyl-CoA, long-chain fatty acyl-CoA, all the same. So when we have a high acyl-CoA levels, and acyl-CoA is important inhibitors of ACC, remember this, right? So under the fasting conditions, high levels of acyl-CoA can inhibit ACC that will lead to decrease lipogenesis, right? And decrease the formation, this reaction decrease the formation of the marino-CoA. So the marino-CoA level is lower under the fasting condition. This will causing the increased CPT1 activity. Does that make sense? Okay. So, and then fatty acids transported into mitochondria, this process is also being increased, speed up, right? And eventually you have more fatty acids being able to enter the mitochondria and go to the beta oxidation. So beta oxidations increase and ketone genesis also increase, right? So again, this is a very important slide. I try to understand all this process, how they regulate it, right? So thinking about the nutrient levels during the fasting conditions and the lipolysis and the lipogenesis process increase or decrease during fasting condition, right? And again, the key point, the marino-CoA and acyl-CoA levels, right? And CPT1 activities are all the key points important, so then hopefully will help you understand the whole, regulatory process of fatty acid oxidation. Okay, so I guess this is all for Okay, now let's move on to lipid metabolism 5 and 6. So cholesterol synthesis and lipoproteins. Right? Cholesterol. So cholesterol is present in two forms: free cholesterol and cholesterol esters, right? So cholesterol ester, basically you have free cholesterol, you have one fatty acid attached to free cholesterol, right, and forming cholesterol esters. The cholesterol present in both tissues and plasma or circulation. Cholesterol has a good side and bad side. The good side, we need cholesterol, right? Because cholesterol is essential structure components of cell membranes, and outer layer of plasma lipoproteins, that which we're going to talk about later. So lipoprotein, the out, layer of lipoproteins also contains cholesterol. Cholesterol is needed for forming the outer layer of lipoproteins, right? And also cholesterol is very important. It's the precursors of or other steroids, like corticoid steroids, sex hormones, bile acid, and vitamin D. They are all synthesized from cholesterol, right? So cholesterol is important. If we need cholesterol for the structural components of the membranes, and also for synthesis of the steroids, right? And bad side of cholesterol, we need cholesterol, but we don't need too much cholesterol, right? So if we have too much cholesterol, cholesterol would be the bad thing for because cholesterol is the components of gallstones, right? And also cholesterol actually involved in the atherosclerosis. So too much cholesterol definitely is not good. And in our body, the cholesterol, half of cholesterol is from de novo biosynthesis. That means our body is able to synthesize cholesterol. So half of cholesterol in our body is synthesized from the de novo biosynthesis. And another half of cholesterol in the body is from diet. So half, right? And biosynthesis, liver, small intestine is kind of important tissue that synthesize cholesterol. That 10% of cholesterol, the synthesized cholesterol, is from liver and small intestine. And of course, the other tissue can also synthesize cholesterol. And cholesterol is synthesized in ER and cytosol. And again, here I wanted to emphasize again acetyl-CoA molecule, right? It's very important. So cholesterol is also synthesized from acetyl-CoA. So acetyl-CoA is also starting molecules of cholesterol. And transport. So cholesterol can be transported between tissues, and two very important the lipoproteins are responsible for transport of cholesterol. These two lipoproteins are LDL, low density lipoprotein, and high density lipoprotein, HDL, right? LDL and HDL are the two important lipoproteins that transport cholesterol between tissues. And synthesis of cholesterol, the process is very complicated, right? I mean, I don't, you guys don't need to remember all the details, right? Just try to remember the starting molecules and the key enzymes, right? So synthesized cholesterol is synthesized from acetyl-CoA. And the first, I just divided by actually 17 enzymes involved and other stuff. So many many steps. So just divide into several steps. So the first step is synthesis of mevronate from 3 molecules of acetyl-CoA, right? And in this first step, the HMG-CoA reductase is the key enzyme for this step and also for the whole cholesterol synthesis process, right? HMG-CoA reductase, again, is the key enzyme for cholesterol synthesis. And also, if you remember, this probably looks familiar, right? When we talk about ketogenesis, these are the same steps or reactions as the ketogenesis, right? See the 2 acetyl-CoA molecule together with acetyl-CoA and to synthesize HMG-CoA, right? And this is the enzyme HMG-CoA synthesis. And HMG-CoA can be converted to the mevronate and by this key enzyme HMG-CoA reductase. So HMG-CoA reductase converts the HMG-CoA, which initially synthesized from 2 acetyl-CoA molecules and 1 acetyl-acetyl-CoA, and break down this molecule to converted this molecule to mevronate, right? And HMG-CoA reductase again is a key enzyme, and this first steps is common steps for both the ketogenesis and the cholesterol synthesis, right? And HMG-CoA reductase can be regulated by substrate and hormones. Again, this is because it's a key enzyme, right? The reason we call this a key enzyme because this activity can be regulated. So the whole process can be either slowed down or up. So, for example, the cholesterol level or bile acid level can inhibit the HMG-CoA reductase, and mevronate levels also inhibits HMG-CoA reductase, which makes sense, right? I mean, if we have high levels of cholesterol, then we don't need synthesize more. So cholesterol level high and can, negative feedback inhibits the HMG-CoA reductase. And also the drug is called statin, is the cholesterol lowering drug, and the mechanism is also inhibits this HMG-CoA reductase. So HMG-CoA reductase is a good drug target, right? So again, this statins is a cholesterol lowering drug, and the mechanism for its effect is to inhibit HMG-CoA reductase and inhibits cholesterol synthesis. All right, any questions? Yes?
Is the red box shown where this is happening in the cell, or is that just?
Oh, this yeah, in the cell. Let me just highlight this key, reaction for the cholesterol synthesis. Okay, so the step 2 is the formation of isopentenyl diphosphate. So mevronate that's synthesized from the step 1 can be converted to isopentenyl diphosphate, and this conversion involves the 3 sequential phosphorylation reactions and the 1 decarboxylation, and requires 3 ATPs, right? And the 3 enzymes three kinases that catalyze the phosphorylation, include the mevronate kinase, phosphomevronate kinase, and the phosphomevronate kinase, right? And then the carboxylase. So you don't have to remember all this enzyme, just give sort of a idea, right? And step 3 is the formation of a screening from isopentenyl diphosphate, and also multiple steps, right? Isomerization, condensation, and reduction. And this steps requires the NADPH. Step 4, the formation lanosterol from screening epoxidase and oxidose screening, lanosterol cyclase, and these two enzymes that involved in this conversion, right? All right, so last step is the formation of cholesterol from lanosterol and isomerase and delta 24 reductase involved in this reaction, right? And 3 NAD pH are needed for this process. So this sort of overall, the process that you started with the acetyl-CoA molecules, right? Again, the substrate or starting molecules for cholesterol synthesis, and involved 17 enzymes and utilizes 8 NADPH and 3 ATPs and to make cholesterol, right? And again, you don't need to remember all the enzymes, the steps. Again, just remember the HMG-CoA reductase, the key enzyme, and how this key enzyme is regulated by substrate hormones, right? And also the first steps, several steps are common step to both ketone bodies and the cholesterol synthesis. And again, the starting molecules, right? This acetyl-CoA cholesterol synthesized from acetyl-CoA, and ketone bodies are synthesized from ketone bodies, right? And fatty acids as well, synthesized from acetyl-CoA. The key enzyme HMG-CoA reductase. Okay, so I guess we'll take a 10 minutes break. Okay. Okay. All right, let's come back. Okay, so now let's talk about regulation of a cholesterol homeostasis, right? Um, we know that keep balance between cholesterol synthesis and disposal is very important, right? We know that we just need the right amount of cholesterol, and too much cholesterol will not be good, right? So keep it, the balance will be very important. So the regulation actually at the two different levels. First is the synthesis. So if we can control the synthesis rate, we can also control the levels of cholesterol in our body, right? And the other side is that the regulation of a cholesterol uptake is also you can control the levels of cholesterol, particularly in the circulation. So let's talk about the synthesis of cholesterol. Okay, so HMG-CoA reductase again is a key enzyme, right, in the cholesterol synthesis pathway. So this enzyme can be regulated by multiple things like hormones and substrates. And for the stimulating factors, which seems like a pretty simple, right? Only two things, hormone, insulin and the thyroid hormone. These two hormones are able to stimulate cholesterol synthesis by increasing the HMG-CoA reductase activity. And lots of the negative regulators, right? For example, starvation basically can inhibit or reduce cholesterol synthesis, and this glucagon, which is a starvation hormone, also can inhibit the HMG-CoA reductase, and glucocorticoid and also inhibits the enzyme activity. And the substrates we talked about it before, right? Cholesterol levels and mavronate, which is intermediates of the cholesterol synthesis pathway. These two intermediates or substrate can inhibit the HMG-CoA reductase when their levels are high. And JAK, statin, is also HMG-CoA reductase is its target and inhibits the cholesterol synthesis. If we wanted to go a little bit detailed in how, in terms of the molecular mechanism, how cholesterol or mavronate, these two substrates regulate HMG-CoA reductase. That involves a transcription factor they call the sterol regulatory element binding protein, SREBP. SREBP is a transcription factor that can control the transcription of HMG-CoA reductase, right? So both cholesterol and mavronate are able to inhibit SREBP transcription factor, then can inhibit transcription of HMG-CoA reductase, right? So just add like one more layer, regulation that HMG-CoA reductase can be controlled by cholesterol and mavronate, can be inhibited by this, right, through this mechanism, the SREBP transcription factor. So again, cholesterol mavronate can inhibit SIEBP transcription factor activities and then control or reduce the transcription of HMG co-reductase, right? Okay, so I think we just finished the cholesterol synthesis pathway, right? Now let's talk about lipoproteins. And we mentioned a couple times before, right? The lipoproteins is very important because lipids, particularly triglyceride, cholesterol, they are not able to freely, transport it to other tissues. So lipoprotein is required to serve as sort of vehicles, right, to can help, the lipids, triglyceride, and cholesterol transport from one tissue to the other tissue. So we have several different lipoproteins, and chylomicron, VLDL, IDL, LDL, HDL, right? These are the common lipoprotein that are present in our body, in our circulation. The chylomicron is the largest, and VLDL the second largest, right? And LDL and HDL are the most abundant in the circulation. So HDL, LDL relatively have higher concentrations compared to chylomicron and VLDL, right? So composition of lipoprotein. So lipoprotein, if we're thinking about lipoproteins, it looks like a ball, right? So the inside the core actually is lipids, and then you have lipoprotein has a surface. So the surface that wrapped the lipids inside of the ball, right? So core basically it consists of a droplet of a triglyceride and cholesterol esters, right? So the inside of the lipoprotein depend on which type of lipoprotein, but in general, lipoprotein core contains the triglyceride and cholesterol esters, right? Again, I mean, depend on the the type of cholesterol. Some cholesterol is called triglyceride-rich cholesterol. That means that in the core has more triglyceride and less cholesterol esters. And the other type of lipoprotein called cholesterol-rich lipoprotein, which means that this type of lipoprotein contains more cholesterol and less triglyceride, right? And a surface usually is the monolayer of the phospholipids, right? The phospholipids, the monolayer phospholipids, the surface, and on the phospholipids layer, there are the free cholesterol molecules, right, integrated in the monolayer phospholipids layer. And also very important, the surface proteins, structure proteins, basically is apoproteins, right? So apoproteins, we have a different type of apoprotein as on the surface of the lipoproteins. So this apoproteins played a very important role in terms of the lipoprotein function, right? So this gives you a better view of the lipoprotein and how lipoprotein looks like. As again, similar like a ball, and inside the core contains the lipids, basically triglyceride and triglyceride esters, right? So free cholesterol basically located on the surface, right? The phospholipid layer. And also on the surface of the lipoprotein, there are different type of apoproteins, right? So again, because of the the lipids are the water, it's not water soluble, right? So you need to be wrapped inside of the lipoprotein that will be able to be transported through the circulation to the other tissues. So apoproteins, we have many types of apoproteins. ApoA 1 to 4, and ApoB 48, ApoB 100, and ApoC 1 to 3, and ApoE, right? And different type of apoprotein, they have different functions. So in general, the function of apoproteins, first of all, apoprotein can serve as the cofactors for enzymes. For example, ApoC2 is very important cofactors for lipoprotein lipase. So that means the lipoprotein lipase activity requires ApoC2. So ApoC2 work together with lipoprotein lipase to hydrolyze triglyceride in the VLDL or chylomicron, right? So just try to remember ApoC2 is important and required for lipoprotein lipase function. Without ApoC2, lipoprotein lipase cannot function well, right? Or cannot function properly. And secondly, apoprotein is also important for structure integrity, can stabilize the lipoproteins, right? And third is some of the lipoproteins, apoproteins, function as a ligand for receptor binding. For example, ApoE is very important for LDL receptors or LDL receptor related proteins. These receptors are very important, right? The receptor is located on the surface of the cells, right? So cells usually be able to take up the lipoproteins, and this receptor can recognize the ApoE protein on the certain type of lipoprotein, right? So ApoE, they help LDL receptors to recognize lipoproteins and to be uptaken into the cells, right? So and then lastly, the apoprotein is also important for help suspension of lipids in the plasma, right? That makes sense. So apoprotein functions. So you need to remember this, cofactors for enzyme ApoC2. It's important for lipoprotein lipase function, and also ApoE as ligands for LDL receptor binding, right? Any questions? Okay. So composition of lipoproteins. So this table just shows us the different type of lipoprotein. There are core lipids, the composition of core lipids, and surface lipids, and also apoproteins, right? For example, chylomicron and VLDL, these are the two triglyceride-rich lipoproteins. That means these two lipoproteins that usually contain high percentage of the triglyceride compared to cholesterol esters, right? So for example, chylomicron in the core, there are 85 to 95% of the lipids are triglyceride, right? And in the VLDL core, 55 to 65% of lipids are triglyceride, right? And LDL, HDL are the cholesterol-rich lipoprotein. That means in the core, they have a higher percentage, the cholesterol, right? A high percentage of lipids, cholesterol in LDL and HDL. And in the apoproteins, different lipoproteins has a different type of apoprotein on their surface. For example, Carmine surface had a ApoB48, ApoC2, C3, and ApoE, right? And if you compare to VLDL, so VLDL basically has ApoB100, ApoC2, C3, and ApoE. So ApoB type of apoproteins are different, right, in between the Carmine and VLDL. So Carmine has ApoB48. Because of ApoB48 is synthesized in the small intestine, and Carmine is also synthesized or packed in the small intestine, right? So that makes sense. You try to connect this, and ApoB100 basically is synthesized in the liver, so this kind of connected to the VLDL. So VLDL surface usually has the ApoB100 and not ApoB48. And LDL is relatively simple and only has ApoB100 on its surface, and IDL is an intermediate density lipoprotein. It's basically is a remnant of the LDL, right? Has ApoB100, ApoE, and HDL does not have ApoB or ApoE, but has ApoA1 and ApoA2. Right? So remnant lipoproteins, because of the purpose of a lipoprotein, again, try to to transport lipids, right? Particularly, specifically, for example, triglyceride from one tissue to the other tissue. The Carmine is synthesized in the small intestine, and the purpose of Carmine is to transport triglyceride synthesized in the small intestine. Initially, this triglyceride is from diet, right? Remember? So, and the purpose of this Carmine is try to transport triglyceride, which initially from the diet to the adipose tissue for storage, or to the liver for further process. So Carmine, once it's transported from small intestine to the adipose tissue, and first of all, we mentioned many times, right? The lipoprotein lipase, which is synthesized from adipose tissue, need to break down the triglyceride in Carmine and release fatty acids, right? So fatty acids then can be picked up by fat tissue and converted back to triglyceride and stored in the adipose tissue under fat condition. So once the most of the triglyceride in Carmine is removed from Carmine, and the Carmine become Carmine remnant, and Carmine remnant eventually is picked up by the liver, right? And VLDL similarly, is packed or synthesized, packed in the liver, and transported triglyceride from the liver to the other tissues. It's also particularly to the fat tissue under the fat condition, right? So triglyceride can also be released from VLDL and picked up by the fat tissue. And similarly, LPL also need to function, right, break down the triglyceride in the VLDL. And VLDL then eventually converted to IDL or LDL. So IDL or LDL is VLDL remnant, right? So the differences between the initial lipoprotein and lipoprotein remnant is the changes in the core lipids content, right? Does that make sense? So remnant, the size is become smaller compared to initial lipoprotein because of lots of lipids can be, already been taken up by the tissues.
Could you repeat that? When they
Yeah.
become smaller? When they become smaller, what?
What's that? Sorry?
Would you repeat the thing with the remnants when they become smaller?
Yeah, the size becomes smaller.
And when they become smaller?
Yeah, because of the lipids, right, been taken. For example, chylomicron goes to the adipose tissue, and adipose tissue take up the most of the triglyceride, right? So then chylomicron become remnant. So remnant is smaller because most of the lipids already been, taken up by the fat tissue. Okay, so yeah, just talk about it, right? The difference between chylomicron remnant in terms of lipid composition. Yes. Okay, so shall we stop here? Okay, so now we have a quiz.